Hello, I'm Shinobu Kitema. I'm this year's APS president. It's my great honor and pleasure to give this year's James McKean Cattell Award to Megan Gunnar. She's a Regents Professor at the University of Minnesota. She's been extremely active in research on adolescents and youth, developmental neuroscience, early childhood, hormones and behavior, infancy, and infant and early childhood mental health. In particular, her work on stress buffering functions of attachment relationship during infancy and childhood is very well known. Her group has found, for example, that friends cannot be an easy substitute for parents as social buffers of stress. Her group is currently working on the imaging paradigm to explore how the changes in social buffering during the puberty period relates to changes in the brain activation patterns during stressors. Big congratulations, Megan Gunnar.
Thank you, Professor Kitayama, for that lovely introduction. It is an incredible honor to be awarded the James Cattell Award. It is also a challenge to think of what to say in 14 minutes to give you all insight into my career. I decided that as it is likely that people early in their careers will be watching these videos, I would take the opportunity to talk through some of the many times that I obtained data that I did not understand at all, and how important those data were to the contributions that I've been able to make to the field. As many senior researchers will tell you, it's not what we found that we expected to find, but what we found that was completely unexpected that were our most important contributions and discoveries. This explains the title of this little talk. When you are wrong is often more important than when you are right. Now, I began my research on stress and development studying monkeys during my postdoc in Seymour Levine's lab. To produce stress in these infant rhesus, we separated them from their mothers. Separation was the stressor. To control for the experience of being captured, which is part of the separation process, we had control infants who we captured, took off their mothers for less than a second, and then reunited. And as you can see in this slide, 30 minutes of separation produced a really large stress response, 240% increase. Those are the bars on the right. I never intended to be an animal researcher. My goal and PhD training was in the studying human infants. So as soon as the salivary assays for cortisol were available, I used my monkey training to decide a study to examine how individual differences in negative emotional temperament were related to how stress reactive babies were. We collected a large number of infants and tested them at 9 and 13 months. At 9 months, we assessed temperament using the Louisville Temperament Assessment Battery, which involved 2 separations, one for 30 and one for 19 minutes. At 13 months, we conducted the Strange Situation Attachment Assessment, which involves 2 brief separations of just a few minutes each. We got the cortisol data back, and I was very disappointed. Here I was going to build a career on studying individual differences in stress reactivity using salivary cortisol, and I could barely get a cortisol increase to the most powerful ethical stressor I could use in the lab. Thankfully, I had already gotten tenure based on the studies I had conducted in the newborn nursery using heel stick blood draws while I was waiting for those salivary assays to be perfected. But still, I was ready to be released from studying newborns, and I could not get a decent cortisol response from infants and toddlers. Now, if a psychological stressor like maternal separations wouldn't do it, perhaps I could fall back on the medical profession and use the shots kids get for their childhood inoculations as my stressor. Again, as you can see in this slide, the data were really confusing. This slide shows the results of a longitudinal study of a large number of infants. As you can see, over the course of the first year and into the second year, the cortisol response to getting held down and injected multiple times was decreasing. In rats, there is this period that's called the stress hyporesponsive period, when the HPA axis that produces the rat version of cortisol becomes very non-responsive. Was it just my luck that this also happens in people? But if so, why wasn't it happening in other primate species? Remember my highly reactive rhesus infants. So I spent a lot of time thinking about the difference between separating baby monkeys and baby humans from their mothers. And I stared at this graph and paid a lot of attention to what might be happening between 6 months when we were getting a response and 18 months. And what I zeroed in on was the parent-child attachment relationship. Remember that control group in rhesus monkeys? We captured the mother and infant, pulled them apart. There was a lot of distress, but the baby monkey was never more than a few inches from the mother, and they were put right back together again. The infants did not show a cortisol stress response. Throughout the well-child checkup, the parent was available to the child. Perhaps attachment was buffering the stress system, blocking increases in cortisol, even if the babies were really upset. If so, that would be pretty profound. It would mean that children came with their own stress blocker. If so, then we would expect that the power of the block would depend on the security of that relationship. And that's what we tested next. And here I finally hit pay dirt. The question I asked was, if I tried to scare 1.5-year-olds by sending in a clown, showing them a clown robot, and having some crazy puppets try to play with them, would the ones who were frightened elevate cortisol if they were insecurely attached to the parent who was with them, but not if they were securely attached? And that's what I saw. The toddlers who were not scared had no reason to activate the stress system, and they did not. Those are the bars on the left of this graph. The same was true about frightened toddlers who were securely attached. But the insecurely attached toddlers produced a clear elevation in cortisol if they were frightened. In fact, their increase was about the same magnitude I'd been expecting based on my monkey research. This finding took me 2 years to get published because the editor, who is still a good friend, could not understand why, if children were frightened and crying and acting upset, why wasn't cortisol increasing? I wasn't able to get into the hypothalamus to find out, but recently Regina Sullivan suggested that the mother's presence, in rats at least, actually blocks the neurotransmitter that stimulates corticotropin releasing hormone neurons in the hypothalamus, and it unhooks the amygdala from the fear distress circuit. In a sense, with the formation of attachment, the attachment figure moves into the child's hypothalamus and becomes a critical regulator of the stress circuit. Just about the time I was finally hitting pay dirt on the role of attachment figures in regulating children's stress biology, another completely baffling set of results landed in the lab. We'd been studying temperament and children's cortisol activity during the preschool day, getting some really nice results in our laboratory nursery school. But my students weren't satisfied. Kids aren't in a half-day nursery school program that much anymore, they said. They're in full-day childcare, and they wanted to see if our results replicated there. To make it more than simply a replication study, which then and now is hard to publish, rather than measuring cortisol once a day for several weeks, as we had done in the lab school, we measured it every morning and afternoon for several weeks with the expectation that we would see more variability in the afternoon and maybe different temperament correlates of the day-to-day variation. But this is what we saw. Cortisol should decrease across the day, but in full-day childcare, it was going up. No way we could have reversed labeled 16 morning and 16 afternoon vials for each child, so the data must be real. But what did it mean? Were we going to have to argue that childcare is stressful for preschoolers? Before making a big noise about childcare stress, my students and I dug deeper. And our first studies actually deepened our concern. This slide shows the difference between morning and afternoon. Negative numbers mean cortisol is decreasing, as is typical for the diurnal rhythm. Positive ones mean that cortisol is increasing. These data come from a combination of several studies, and what you can see is that for children in the infant room in these childcare centers, cortisol does decrease over the day. Children eat and sleep pretty much on their own schedules. There's lots of one-on-one interaction, and cortisol was going down. But once the child moved into the toddler room, where everyone eats, sleeps, and plays at the same time, among the younger preschoolers, the cortisol rise was really large. Among the older preschoolers, the increase from morning to afternoon was smaller, but still significant. And then among the 6-year-olds and older children, cortisol decreases across the day were the same as if these children were at home all day. These types of data started a cottage industry of many folks testing cortisol at childcare. It was actually rather embarrassing to have started such a craze. But what became clear is that the quality of care predicted how much of a rise in cortisol childcare produced. But it did not answer the key question: does it matter? Over and above childcare quality are these elevations in cortisol every day the child is in care, having an effect on the children's development. I knew it would be irresponsible of me to leave this area until I had that answer. This slide shows what we learned. First, we saw that children in family daycare homes so that we could look at a lot of different settings. Second, we saw them at time 1 and again 6 months later at time 2. In between, we brought them into the laboratory to objectively assess emotional temperament. My hypothesis was that elevations should have effects primarily on children who are behaviorally inhibited or shy. The combination of more social anxiety and experiencing elevated cortisol each day should increase the reactivity of fear circuits and enhance internalizing problems. And for once, I was right. This is what we found. These data were analyzed continuously, but I'm graphing them here dichotomously. Cortisol should decrease normally over the day or rise. Children can be high on behavioral inhibition or not. Cortisol behavior at childcare was not associated with internalizing problems for those who were lower on behavioral inhibition. For those higher on behavioral inhibition, being in a childcare home where you experience normal decreases in cortisol over the day was associated with fewer than normal internalizing problems, while being in a home that produced increases over the day heightened your internalizing problems. So the answer is nuanced, as many things are. If you're a shy, inhibited child going to a high-quality childcare where you do not elevate stress hormones each day, this seems to be beneficial in reducing anxiety and depressive symptoms. But for this type of child, being in a childcare that is more stressful may be counterproductive, increasing anxiety and depression. We found nothing negative for the children who were not shy and fearful. The last area where I was wrong led to a whole change in our understanding of stress, and it came from a trip I took to Iași, Romania. I was invited to go and collect cortisol levels on children in an orphanage. Everyone, myself included, expected that since these children did not have attachment figures to regulate stress, they would show elevated cortisol levels. But when we got the samples home and assayed them, I was again staring at data that I did not understand. Compared with children who lived with their families in Iași, the children in the institutions had low cortisol levels early in the morning when cortisol should be high, and they stayed relatively flat across the day. This pattern is associated with hypocortisolism, not hypercortisolism. We saw this in 1995, and soon we're replicating the finding as I was leading a consortium of researchers. We were replicating the finding for maltreated children entering new foster placements and for children with stunted growth due to chronic stress. By 2001, we were sure enough of these findings to publish the first paper alerting developmental psychology and developmental psychobiologists that, unlike in rats, chronic stress in childhood produces hypocortisolism. Not only has this paper been cited over 1,000 times, but the basic finding has been replicated many times and shown to be true in several recent meta-analyses. Whether hypocortisolism is more closely aligned to deprivation than threat is currently being worked out. But the point is that once again, being wrong opened the door to a whole new view of the psychobiology of stress in human development. Now, I hope I've encouraged you not to despair when you fail, to be right. Instead, if you've designed your study well and you believe your data are solid, don't try to shove it into your pre-existing hypotheses. Pay attention to what your data are trying to tell you and dig deeper. And again, I want to say how honored I am to have received this award, and I thank you for your attention.